Thank you for joining us today, everyone. As a reminder, all GSA online events are covered by a code of conduct, and the link for that is available in the chat. Hugo, please go ahead and start when you are ready.
All right. That was a short introduction. All right, can you see my screen?
Yes, we can.
All right. So today I'm going to be talking about tackling human gene function in model organisms. And this is a seminar or a series of seminars. This is the first of a series of seminars sponsored by the Genetic Society of America. And so the goal of the seminar is, and the research that we do, is to advance both basic science and medicine by studying human genes associated with diseases in model organisms. So that will be the theme of the seminar series. And the idea is to promote collaborative efforts among geneticists and other scientists. First, to help in the diagnosis of rare disease. Second, to elucidate the molecular and pathogenic mechanisms of disease using model organisms. And to help and participate in the discovery of drugs using model organisms. And finally, to promote the discovery of function of genes that have been poorly characterized in model organisms. So a basic introduction, which are really in essence trivial facts, but are kind of important. Obviously, the main tenet of this research is that genes are conserved in evolution and their function is conserved. That is obviously not always true, but as a proof of principle, I would say that in yeast, worm, and flies, about 50% of the genes can be rescued by the ortholog, the human ortholog, using a single human cDNA. So this argues that a lot of the genes are functionally conserved and can perform their function in the model organisms. Obviously, a main advantage of invertebrates is that there are fewer homologs than in the vertebrates. And for many genes, there is also a real ease and speed of genetic manipulations when compared to vertebrate. And that is really important because it makes it fast, cheaper, and easier to manipulate the organism. Obviously, a main advantage of vertebrates is that it's much easier to compare and interpret phenotypes associated with genes in human conditions. So the point here is that many different organisms can contribute to these studies, including all the typical model organisms, but also other model organisms, as long as they can be genetically manipulated in an easy and straightforward manner so that they can also contribute to the study of human disease. A few words about technology, because technology is really a driving force here. The idea is that you have to be able to quickly, efficiently, and precisely determine changes or create changes, and that's going to determine the success of the cross-species approach. We use typically four main approaches in this research. First, we create loss of function using RNAi or CRISPR. Key is to try to rescue the mutant phenotypes, preferably with human cDNAs, but this can be done in other ways, as I'll expand about. One likes to model point mutations, if possible, that are found, the variants that are found in the patient. But one can also use overexpression studies, and I'll try to present examples of each of those to show you that they all can be informative. As already mentioned, the different technologies in different model organisms are vastly different and can obviously impact the choice, what reagents are available, what mutants are available, etc. And obviously the nature of the question, what is your primary interest, will also determine what model organism to use. So a few strategies with respect to creating loss of functions of phenotypes through RNAi or CRISPR. You want to define the phenotype of the ortholog in the model organism of the human gene, and it obviously makes sense to pick the best ortholog in that species based on the diop score. To avoid species in which the human gene is duplicated, because then you have to make double or triple or other more sophisticated knockouts and spend more time and energy on it. We typically use RNAi through ubiquitous or tissue-specific expression. And that has been very useful. Even more useful has been the creation of CRISPR mutations, either by creating deletions or insertions, or by inserting the Gal4 technology, which is especially powerful in the fruit fly and is now being explored in other model organisms. And so I'll briefly expand upon what we do and why we like this approach. So expressing the human gene, if possible, to rescue the mutant phenotype can be very informative and has been done by first knocking out the gene by RNAi or CRISPR and then to induce the expression of the human gene and see if you can rescue the phenotype. A few words about the technology that we use in the fruit fly that has been particularly useful is the CRISPR technology, in which we insert an artificial exon that contains the T2A Gal4 poly-A tail. And the idea here is by creating this artificial exon that has a splice acceptor, during transcription, there is an arrest at the poly-A tail, so this about 90% of the time will lead to a truncation of the gene, and most of the time will create an allele. You can then obviously have a splice donor that will go to the splice acceptor and integrate this exon into the transcript, and this exon contains the T2A Gal4. And the idea is that when during translation it hits the T2A, it will initiate and just produce the Gal4. And the Gal4 will there be produced in the right tissue and at the right time. That is, it will mimic the endogenous gene expression pattern. This technology has been changed now. I show here where, you know, we put in 1 kb homology arms, but we now use 100 base pair homology arms, and we've shown that this is now very efficient. And if you're interested, I refer to this Kanka et al. Paper, where we have about 80% efficiency. So we can make a piece of DNA that we order from a company and insert it, and about 80% of the time we get success with this approach. And so this is a very relatively simple and powerful approach. Why is this so useful? Well, the first is you can insert a cassette that, as I mentioned already, will create a gene disruption. And this is very efficient. 90% of the time we create an allele. The second issue is that you can cross your Gal4 strain to UAS flip. So now you will excise the cassette that you've inserted. And by doing this excision, you revert the phenotype, and therefore you've demonstrated that there's no other second site mutations on that chromosome, or that there is no background issue. So you know that your experiment is clean. The third issue is that you can insert, because they're at P sites, other cassettes. You can create artificial exons by removing the original T2A Gal4 and replacing it, like is shown here, with a GFP or multiple tags that are attached to the GFP, and create an artificial exon that has a splice acceptor on one side and a splice donor on the other side. And by creating this artificial exon, you now fuse the coding regions here to this GFP. And in about 70 to 75% of the cases, this will revert the phenotype that was associated with this cassette. So now you have a tagged protein, and it's functional, and you can do very simple but very nice experiments. You can do protein localization studies, you can perform immunoprecipitations and find interacting proteins, and you can use systems to degrade this GFP using the degradFP system, and therefore remove the protein in any tissue at any time. So this is providing another nice tool to study the function of your gene. Finally, you create a GAL4 insertion, and the GAL4 insertion can now be used to rescue the mutation that you've created by just crossing the GAL4 to either a fly cDNA, and again, that shows that you've now induced other mutations on the chromosome. You can use it to humanize the fly and drive a human cDNA. And you can, if that's the case, and the human cDNA rescues the fly gene, you can test variants that are found in patients and see if they rescue or not. Finally, you can use a fluorescent protein and determine where your gene is expressed. We typically use a nuclear fluorescent protein so we can define the cells that express this gene. So it's kind of an integrative approach that allows many different manipulations, including protein localization, gene expression, rescue, and various biochemical experiments. As I mentioned, we like to humanize the flies. That is, we like to cross the mutations that we've created with the GAL4 to human cDNAs. These human cDNAs obviously need to be made available, and we decided to collaborate with Sue Selnicker and Shinya Yamamoto at Baylor to create a library of about 9,000 human UAS cDNA constructs. And those can also be used in other species, by the way. They're deposited in the DGRC in Bloomington, and they allow, on the one hand, the rescue of the T2A GAL4. They can also be used to rescue RNAi, because obviously the RNAi is not effective on the human gene, and the human gene can therefore be used to rescue RNAi phenotypes. And they can also be used to assess if there is a loss or a gain of function or a dominant negative in the model organism. So it provides us with a nice set of tools. We're about halfway through this project. We've generated about 5,000 or 6,000 US human cDNAs, and we've made transgenes for about 4,000 of them so far. And this is in collaboration with Toshi Takano Shimudzi at the Kyoto Stock Center, who also performs a lot of injections and makes these strains available in the Kyoto Stock Center. All right, what other issues do we need to talk about? First, the rescue of mutant phenotypes. As I already mentioned, you create these human cDNAs and you try to rescue their phenotype. This doesn't work all the time. You pick one long open reading frames of cDNA that encodes a long open reading frame. We call that the reference, or wild type human cDNA. And humanization doesn't work all the time. The typical success rate is about 50% if you put the conditions rather stringently. It's about 70% if you're not too stringent and you're just screening for some phenotypes, not necessarily a full rescue. If that fails, and there's many reasons, you may not have picked the right cDNA, there may be issues with codon issues, you can then step over and use the model organism cDNA to rescue and model the variants in the model organism cDNA. The typical success rate of that is 70%. Again, many genes encode many different isoforms, and so you don't always get rescue when you do that. If the rescue does not succeed in either the model organism or with the human cDNA, we typically step up to another alternative, and the alternative here is to use genomic rescue experiments based on BACs. We made BAC libraries in fruit flies, and they're available in many other species. And you can create transgenic animals that carry the BAC, and this is almost invariably will lead to a 100% success rate because you include the open reading frame with regulatory elements, and that typically seems to work. You can then model through recombineering your variants in the back that rescues. Overexpression studies are also used quite a bit. That is, if the cDNA strategy and the humanization fails and the model organism gene fails to rescue, you can step over to another strategy and do overexpression studies. Overexpression of quite a few of the human genes causes a phenotype. And if the variant found in the patient does not cause a phenotype, then we interpret that as a loss of function. Sometimes the phenotype of overexpression of the gene is creating a phenotype, but it's not as severe as the reference cDNA, so then we have probably a partial loss of function variant. And sometimes the variants cause the same phenotype as the reference when overexpressed, and then either it's not affecting the protein, or it may be a gain of function mutation, and that obviously needs to be explored further. Overexpression of the reference human gene often does not cause a phenotype, but then when we test the variants that are found in the human, they sometimes cause a phenotype. So that suggests that maybe there is a gain of function or a dominant negative function associated with that variant. And then finally, the variants observed in the human do not cause a phenotype. Then obviously, if the overexpression of the wild type and the mutant neither cause a phenotype, there is essentially no conclusion. All right, finally, modeling small genetic changes, which refer to single amino acid changes, small deletions, insertions, etc., are typically done using CRISPR technology, and they either are done in the genome, or if you do a humanization experiment, you want to generate these mutations in cDNAs for your rescue experiments, or if the human gene doesn't rescue, you can go to the backs in the model organisms. And we typically do this in flies when the genes are large and cDNAs are not available or rescue fails. So, you know, as I said, the rescue with backs almost invariably works, and therefore, if the genes are conserved, they provide another avenue. All right, let's go to a first scenario where, driven by curiosity, you know, numerous model organism researchers in all of the fields that we typically work on as geneticists screen and identify novel genes. And very often, that the model organism researchers will pursue that research, but fail to connect the human geneticist. And the human geneticists perform, in essence, genetic screens to identify the cause of numerous rare human disease. So very often they may have in their database mutations in the genes that you're studying in the model organism. And the idea of the seminar series is to try to connect the model organism researchers with the human geneticists, and then ties them to explore databases so that they can make these connections. So if both groups have identified orthologs in the same gene, how do you connect with each other and maybe impact diagnosis if the gene is not known to cause a human disease? And obviously basic biology, because a lot of the genes that they identify may not have been studied very well. And that's why they have no idea if the gene causes the disease or not. I'll take an example from my lab where we did this, and we started with an unbiased genetic screen in Drosophila. We performed an EMS mutagenesis and created 6,000 or 7,000 stocks that are homozygous lethal. These flies carry an FRT on the base of the X chromosome, and so you can induce in heterozygous animals mutant clones in the eye. And therefore, we performed electroretinograms to screen for mutations that cause a progressive loss of the ERGs. And we also, at the same time, performed a screen in the thorax where we created clones in the thorax and screened for bristle loss. So obviously, the bristles are the peripheral nervous system of the fruit flies, and this is also the peripheral nervous system. But it allows us then to identify genes that cause phenotypes and map these genes. What we did is we identified 700 mutations and did complementation tests on the X chromosome, identified 165 genes. And we did that by doing the rescue, because they're all homozygous lethal, by doing the rescue with duplications. And then we sequenced all these flies and identified this list of genes that is listed here. And so here we have a list of a fly gene and a human gene. And what you can see is that many of the fly genes that we identify have human homologs. Only maybe 12 genes in this whole list don't have human homologs. So this helps us then to make connections. And the question then is, what of these new genes that we've identified that have not been associated with human disease yet are causing potentially a disease? So when we published this paper in 2014, about 30% of the genes, those that are labeled in red, were associated, 30% of the genes that are labeled in yellow were associated with human disease. And in the last 6 years, this number has more than doubled. And that shows you how intense the effort is to identify new human disease by the human geneticists. So when we had the 60% of the genes or 65% of the genes at the time that were not associated with human disease, we contacted the Center for Mendelian Genomics at Baylor College of Medicine and asked them if they could screen their databases and find potential families that have been sequenced where they've identified a gene that they think may be responsible for that disease. So the Centers for Mendelian Genomics are centers in Johns Hopkins and Baylor. That's a joint venture, the University of Washington, Yale, and Broad Institute. And one of the points I want to highlight is that Mendelian gene discovery is really going at a very fast pace. And if you look at this commentary from Bamshat, you will read that finding the genetic basis of 6,000 to 13,000 Mendelian conditions yet to be delineated will require both technical and computational innovations and will rely for a very important extent on data sharing. And so I think this really is what this seminar is all about, is how do you make connections? How do you define, what the function of your gene is, and how do you connect that to human biology? So the Center for Mendelian Genomics is focused on sequencing patients and unaffected individuals, but mostly patients, to discover the genomic basis of Mendelian disease. And then their goal is to disseminate this knowledge either through publication or through posting the sequences in databases where they can be queried, ClinVar and dbGaP. And so they create a candidate gene list that you can look up, and this seminar will be posted, so you can look up these databases and see if any of the genes you're interested in fall into this or present into this database. You can also use GeneMatcher and Matchmaker exchange, which I'll talk about briefly. But with respect to the CMG, Baylor Hopkins has sequenced about 10,000 exomes. Baylor Genetics has sequenced about 12,000 exomes. And I list the names of the people here that you can contact at Baylor or at Johns Hopkins, where you can present a set of genes and they will query the databases and they will send you lists of the variants that they've identified in these genes and what the phenotypes are that are associated with these genes that are not yet known to cause a human disease. So you can participate in the discovery. In our case, when we submitted this 165 genes, we got 6 hits. And I just wanted to kind of briefly talk about one of those hits, ANKL2 mutations. So there were 2 affected individuals here in this family. Both the father and the mother are a carrier of a variant in this gene ANKL2. And the ANKL2 protein is shown here, and one of the mutations is here, and one causes a truncation. And the brother, who survived for 6 or 7 years, had a very severe microcephaly, as is shown here, and a very severe loss of brain tissue. So at birth had a severe microcephaly and failed to thrive and essentially stayed as an infant for many years before he died. So the idea was ANCL2 was identified in the screen as a gene that affects the development of the peripheral nervous system. What about the central nervous system? And as is shown here, when we looked at the third instar larvae of the ANCL2 mutations, we noticed that these ANCL2 mutations have a small brain compared to the control, and then we expressed the human ANCL2 and restored not only the brain size, but these animals are lethal, and these animals are viable and survive. And detailed analysis showed that they have fewer neuroblasts, that the neuroblasts divide less frequently, and that there is increased apoptosis. So this provides already compelling evidence that the gene ANCL2 is indeed the cause of this microcephaly. Now, interestingly, the microcephaly that was observed in the ANCL2 patients is very similar to the congenital Zika syndrome microcephaly. Not all microcephalies are this severe and express such a similar phenotype as is shown here. And interestingly, we were contacted by Nevin Krogan at UCSF and Priya Shah at UC Davis because they found that a key Zika virus protein binds to ANCL2. And there will be a seminar in the same series here because I think this is a really nice example of a genetic disease that's really rare, having a much bigger impact because it is the target of a viral protein that causes Zika syndrome. And so Priya Shah and Nicole Link will present their data that they've accumulated over the last few years related to unraveling the links between hereditary and viral microcephaly. So I encourage you to participate and listen to that webinar on November 1st. And I want to switch to a second scenario that is pretty common now. Rather than being driven by the model organism researcher who is interested in identifying human disease, human geneticists perform genetic screens to identify the cause of numerous rare human diseases. There are huge databases of potential variants that may cause human disease. And if they can sequence the parents or other members of the family, they typically end up with one or a few candidates that they think might be the cause of the disease. Now, the orthologs of these genes can sometimes are well studied. Let's say you identify a mutation in notch or wingless, or have not been studied at all. And I think this leads to a kind of a bit of a different scenario. In scenario A, where the genes have been well studied, the physicians can team up with labs that are really well versed in studying these pathways, and in which these orthologs have been studied in more model organisms, or at least there is an inkling that they are playing a role in that pathway. And that is, again, possible through model matcher and gene matcher that I'll expand upon, or international efforts like the RDMM in Canada, where physicians can team up with model organism researchers, because the model organism researchers have listed genes that they're potentially interested in, and therefore the physicians know that they can kind of dovetail with that research that's already occurring in that lab. In scenario B, they directly contact model organism researchers who will tackle the genes, even if they're unknown. And that model is the model of the Undiagnosed Diseases Network that we're participating in. And I think, there are such kind of strategies that are being developed throughout the world, in Japan, in Australia, in Europe, etc. And so how do you go about when, contacting a model organism researcher who's not particularly interested in your gene of interest? And so how do you do that? And how do you impact, again, diagnosis and basic biology? The first one is Gene Matcher, and Gene Matcher started in 2014 and is a freely accessible website designed to enable connections between clinicians and researchers. And we will again have a seminar about this Gene Matcher's approach as well as the model matcher, which is much more recent and was developed by Shinya Yamamoto more recently, where the idea is that if you want to collaborate or find out if someone else is working on your gene of interest, you can log on to Model Matcher, put in your data, and it'll make a connection for you with those researchers that are interested in the same or similar genes. So here, again, there will be a GSA seminar matching interest across research teams for Gene Matcher and Model Matcher from John Hopkins and Michael Wangler at Baylor College of Medicine. Another important set of databases is Matchmaker Exchange. And this is really a complex network of databases that you can query. And for those of you who are interested, I encourage you to go to Matchmaker Exchange because you can find a lot of information there. And if you know how to mine these databases, you can clearly participate in the discovery of quite a few human diseases if you're interested. Now, I want to briefly talk about the UDN, which is the second system where there is a structure, and that structure, as I said, is being developed in the United States, but also internationally in many different countries. And in the United States, the Undiagnosed Diseases Network is a network of clinicians and scientists who work together to diagnose rare disease. In the United States, the UDN has accepted 5,000 patients that have applied, and not all of these are pursued, but a substantial fraction, about a third to half, are pursued in the Undiagnosed Diseases Network. And they're assigned any of the clinical sites that are shown in those red states here. There's 12 clinical sites, and they get a free workup that is essentially, they do a phenotypic analysis. They look at the patients and do a whole series of tests to try to see if they can diagnose them. And if they fail, then they will sequence their genomes, which by sending the DNA to Baylor College of Medicine, where the sequencing is performed. And they will typically sequence a trio or quattro, that is, they'll sequence the kid and the parents. 65% of the submissions here are children. And so we can easily identify the parents and try to determine with a trio what are all the candidate genes that might be the cause of the disease. So after the sequencing and the analysis of the sequencing, they typically come up with 1, 2, or 3 variants, and those are then being submitted to the model organism screening centers, of which there are 2. There is a venture between Baylor and the University of Oregon, and there is one at WashU now. And we team together, and we perform a deep bioinformatic analysis of the mutations, the genes, and the variants through this program called MARVEL. And MARVEL integrates data from human and model organisms and allows us to figure out what the best species is to model the disease. And we have 3 species: C. elegans, Drosophila, and zebrafish. Zebrafish are being modeled at WashU and at the University of Oregon, Drosophila at Baylor, and C. elegans at the University of Washington. And so we all work together to try to see what the best strategy is to model the disease. And a brief word about MARVEL, and again, there will be a seminar about MARVEL at August 23 this year, mining genetic and genomic data across model organisms and human. That is to help model organism research understand how you mine all the genetic information in humans and model organisms, and help you understand how you need to prioritize genes that may cause a human disease. And so MARVEL has been very useful for us, and it kind of requires a tutorial. There is another aspect to MARVEL, that is, MARVEL is now being used to develop artificial intelligence, where the idea is that when the physicians submit sequences from parents and children, that it will query together with the phenotypes that are being submitted, which gene might be responsible and should be modeled in model organisms. And they've become very proficient now at doing this. And Zhangdong Liu will explain what's happening with respect to artificial intelligence and more. Another strategy and database is to mine the information that's present in the Alliance of Genome resources, which is created by all the model organism databases, from yeast to flies and worms and mice, and fish and rat. And the idea here is that people can mine this information, and that the information that's gene-centric and shared between all the model organisms can be efficiently mined in the Alliance of Genome Research database. And so this is essentially what they've come up with in the last 4 or 5 years with respect to extracting as much information from the model organism databases and putting it all together in a coherent set of data that can be searched. And Monty Westerfield and Paul Sternberg will give a webinar about mining model organism data in the Alliance of Genome Research portal, and that will occur on September 20. Finally, there is another initiative called the Monarch Initiative, where they try to connect phenotypes to genotypes across species. And that is also a valuable database to extract information from and try to determine, if the gene that is being identified in a human may cause a human disease. And so that's the Monarch Initiative, and Melissa Handel will present a talk on the cross-species phenotype comparisons for disease diagnosis and discovery, again in a webinar that will occur on October 4, 2021. All right, let's now turn back to what we've done with the Model Organism Screening Center, and the idea here is to give you a few examples of what has been done in the last few years with respect to the discovery of these rare human diseases. Most of the time, we start with one patient and one variant or two variants if the mutation or the disease is recessive. And the humanization is, in essence, the most productive, but also the most straightforward. And I'm not going to cover any of the examples there. You make a mutation, you rescue it with the human gene, you test the human variants, you show the human variants show a different phenotype than the wild type, and you try to participate or have presented evidence that it's the cause of the human disease. The other cases are a little bit more tricky, and the first one that I'll cover is ACOX1, where we studied the variant in the fruit fly and try to understand the molecular mechanisms that are at the root of this ACOX1 mutation that are at the root of this disease. I'll briefly talk about an overexpression strategy in the fly to show you how we can use overexpression experiments to determine the nature of the allele. I'll talk about loss of function studies to identify similarities between the fly and the patient and using IRF2BPL. And finally, I'll talk about a zebrafish story that was performed at the MOSC in the University of Oregon about Yippee-like 3. And so I'll give you a few short stories which serve as examples as to what you can expect if you start engaging in this type of research. ACOX1 was a gene that we showed that it affects the very long chain fatty acid metabolism and causes the demise of glia. And this was, again, a collaboration between teams at WashU, Baylor, and the people that are involved in it, especially Lok Chung in my lab, did most of this work. So what is the role of ACOX-1? Not many of you have probably never heard of ACOX-1. I certainly didn't. It's a peroxisomal enzyme that is involved in the beta oxidation of the very long chain fatty acids. So you know that every cell has peroxisomes, and very long chain fatty acids are synthesized on the basis of long chain fatty acids, and they're elongated through this protein LOVL1. And when they're longer than 22 Cs, then they are modified and imported in peroxisomes. And the first step of the beta oxidations of these fatty acids to make acid CoA, which is going to be transported to the mitochondria for energy production, occurs in the peroxisomes. So this is the rate limiting step of the beta oxidation in the peroxisomes. And this beta oxidation of the peroxisomes leads to very high levels of production of H2O2. So this is potentially toxic, and therefore catalase in the peroxisomes will ensure that there is no high levels of H2O2 and that there is a minimum of ROS production. Obviously, loss of ACOX-1 causes an ACOX-1 deficiency, and when there is an ACOX-1 deficiency, there is an accumulation of the very long chain fatty acids. And an accumulation of the very long chain fatty acids is very toxic. These children are very severely affected, and they typically die within 3 to 4 years after birth. They're very severely affected. There's another disease, ABCD1, that is called X-linked adrenoleukodystrophy, which is a mutation in this ABCD1 transporter that takes the very long chain fatty acids and incorporates them into the peroxisome. So the UDN submitted a patient with a de novo mutation, and this is different than mutations in patients that were published before, because this is an autosomal recessive disease. It's the loss of ACOX-1 causes an accumulation of the very long chain fatty acids and is associated with a severe inflammatory response, a white matter demyelination, an ataxia, and as I mentioned, early death. This patient, though, that was submitted from WashU, called Mitchell, had a disease onset at the age of 12, died by the age of 19, had a progressive disease course, had no increase in very long chain fatty acids, no inflammatory response, no white matter demyelination, but became ataxic when he was 12. So this is obviously a very different progression than what is defined for the loss of function, suggesting that maybe this point mutation causes a gain of function mutation. In fact, we found a second patient in Korea with the same point mutation and a similar progressive disease with, again, no accumulation of very long chain fatty acids or inflammatory response and a white matter demyelination late. And this is also what happened in this patient. He also developed a late white matter demyelination. So same mutation. In fact, there's now 7 patients with exactly that same amino acid change. And they all show a very similar phenotype. So Locke thought this might be a gain of ACOX-1 function. And to make a long story short, we showed that ACOX-1 was expressed in glial cells and almost not in neurons. And that all the phenotypes that were observed in the patients could be attributed so far to 2 cell types, the glia and the immune cells. And in the ACOX-1 deficiency, there is a loss of ACOX-1. And there is an altered very long chain fatty acid metabolism, and there is an immune cell activation, and the immune cells seem to attack the glial cells. And a drug that we tested in fruit flies, bezafibrate, which suppresses the production of the very long chain fatty acid, has a very beneficial effect in the fruit flies. But in the absence of this drug, there is an axonal degeneration because the glial cells that surround the axons, the wrapping glia that surround the axons, are affected. In the variant that was identified in the patient, we showed that it was a gain of function mutation. And this gain of function leads to very elevated levels of H2O2 production and elevated levels of reactive oxygen species in the glial cells. And this also leads to glial cell degeneration for a very different reason than here. And an antioxidant, AD4 or N-acetylcysteinamide, is a very potent suppressor of this gain of function mutation. So very different mechanisms, very different drugs. And in fact, this drug, the antioxidants, are currently being applied for at the Food and Drug Administration to be used in these patients that have been identified so far. Now turn to overexpression studies. I already mentioned that overexpression studies can also provide some information. And this was a study that was done by Lindsay Goodman in the lab in collaboration with the Duke team from the Undiagnosed Diseases Network, and relates to a transportin. And again, de novo variants in transportin 2 are associated with developmental delays and alter the function of the protein. And this was almost entirely done using overexpression studies. So we started with one patient from Duke and then posted on Gene Matcher and Matchmaker Exchange, et cetera, and eventually identified 15 patients with de novo variants in TNPO2. These variants spread throughout the protein that is shown here. This protein is involved in the import of cargo into the nucleus. So transportin has a run-binding domain, an acidic loop, and a cargo-binding domain, as shown here. It binds cargo and will import that cargo into the nucleus, where the run GTP will bind to this run-binding domain and release the cargo into the nucleus. So because there's 15 patients, we identified a lot of different variants, and many of these variants are suggestive of causing a severe phenotype. What are the phenotypes associated with these patients? First, global developmental delays, and that is observed in all the patients. Dysmorphic features, behavioral features, feeding deficit, ophthalmological features, and muscle tone deficit, which is found in all vast majority of the patients. And then movement disorder, seizures, microcephaly, and brain abnormalities that are found in about 30 to 40% of the patients. So obviously, given that we have so many patients and that there is a clear similarity, this is very suggestive that these mutations that we've identified are the cause of the disease. But in the beginning, we didn't obviously know that. And so we had identified previously in the Yamamoto screen a mutation in transportin in the fly and shown that it was homozygous lethal. And we induced and found that, you know, there was a loss, a similar loss of function with similar phenotypes when we induced a CRIMIC allele or looked at a deficiency. And all of these mutations could be rescued with a genomic rescue. So we induced clones in the wing, and I'm not sure what happened here because the clones that we induced in the wing caused a blistering phenotype and pretty obvious phenotype. Obviously, when the mutation is homozygous lethal, it's simpler to do a specific knockout in specific tissue. And we did that in the eye and the wing and showed that there were phenotypes associated with the loss of this protein in the eye and the wing. Now, the human gene did not rescue any of the phenotypes that were associated with the gene. So we had to resort to another strategy to try to define the function of those variants. And what Lindsay did was use the eyeless Gal4 system to overexpress this gene in the eye and showed that the reference cDNA, when overexpressed in the eye, causes a strong phenotype. In fact, this phenotype is quite similar to the loss of function phenotype of the fly gene in the eye. But then notice that here she tests the same cDNA with a single amino acid change. And in one case, there is almost no phenotype associated with it, suggesting that this one is toxic, and this point mutation removes this toxicity and therefore is likely to be a loss of function mutation. But another mutation in the protein still causes a very similar phenotype to the gain or overexpression of this phenotype. Interestingly, the same was observed in the wing when you drive with a wing-specific driver, the UAS, and look at the reference cDNA. You see clearly this kind of blistering phenomena as well as some subtle phenotypes in the wing. Again, when you overexpress this mutant variant, you see very subtle defects in the wing. Most of these are not. The defects are not what is observed when you overexpress the protein. But this mutation here, the same mutation as is shown here, when overexpressed in the wing, causes a very severe wing phenotype. So this argues that maybe this mutation is a loss of function mutation, and this mutation may be a gain of function mutation. And by doing this kind of systematically and looking at a whole slew of variants, what Lindsay found was that these mutations here that affect the RAN binding domain seem to behave as gain of function mutations. The mutations that are in this area here in the cargo binding domain are loss of function variants, and the mutations that are in the flexible loop of the protein kind of behave different in different tissues and show kind of a loss of function, but that loss of function phenotype is tissue specific. So what the study shows is that, hey, there's a lot of patients that we identified, and when we modeled these patients and looked at the amino acid changes in different domains, they seem to cause different phenotypes. And so we infer from this that mutations in the RAN binding domain may cause a gain of function phenotype, whereas in the cargo binding domain, they may cause a loss of function phenotype. And in the flexible loop, they may cause a variable phenotype. So based on this, then, I want to step back and go to the two last stories that I wanted to cover. The first one is IRF2BPL, which refers to this gene here, and the story in zebrafish, which refers to yippee-like protein 3. So loss of IRF2BPL impairs neuronal maintenance through excess Wnt signaling. And here what I show is that you see in patients what's happening with the brain of this particular patient whose MRI was taken at 7 years, 13 years, and 20 years. So this patient had a de novo mutation in IRF2BPL, and you can see the very severe loss of brain tissue that occurs over this time span. This work was done by Paul and Depdeep in my lab, and is a collaboration with Duke, with Lauren Penna and Vandana Sashi. So this is a very severe disease in children, just like all these other diseases that are already covered. They develop normally till the age of 3 to 7, then become ataxic, very quickly wheelchair bound, and eventually completely unresponsive or tube fed, and eventually die. The gene orthologs are really poorly characterized for this gene. It's been shown that in cancer, they may affect Wnt signaling when lost. And the UDN submitted the first patient in 2017, and now there are 27 patients. And the mutations of those 27 patients are shown here. This protein has a DNA binding domain with a zinc finger, has a nuclear localization signal, and a ring finger. And so the disease by the OMIM database is called NEDMUS, for neurodevelopmental disorder with regression, abnormal movement, loss of speech, and seizures. Not all the mutations in this gene cause NEDMUS. The ones in red here are shown cause NEDMUS. The ones in blue, which are much more subtle, cause an autumn spectrum disorder, and some of these other more developmental disorders. But the typical NEDMUS phenotype for these de novo mutations that cause a truncation in IRF2BPL are disease onset typically between 3 and 7, but as late as 23. Survival is unclear, but several already have died. The ataxia is an obvious phenotype early on. There's severe loss of milestone. There is seizures, and there is a very severe brain atrophy, as you've seen in one of the longest living patients. So this is a pretty devastating disease. The homologue of IRF2-BPL is called PITS in Drosophila. So in Drosophila, there's only one gene, PITS, but in humans, there's three. There's IF2-BPL1, BP1, 2, and L. The loss of PITS in Drosophila is embryonic lethal, and that RF2-BPL truncations, when modeled in fruit flies, act as loss of function. So what Paul and Debbie did is they started working on this gene and trying to figure out, what if we reduce PITS at a later stage or in specific tissue and determine what the phenotypes are. And so they did this, and they found that when you remove through synaptobrevin and the degrad FP system, so here the PITS was labeled with GFP, and the degrad will degrade the protein. So you can put the flies at 18 and not have any induction of the degrad FP, but if you bring them to 25 or 28, the degrad FP will bind to the GFP and degrade the PITS protein. And you can see if you do that for 25 days, you can see that start developing climbing defects. And we then sectioned and did transmission electron microscopy on the wing nerve, the anterior wing nerve, which has a couple of hundred neurons, and therefore in this nerve has a couple of hundred axons. And you can see what happens after 25 days when you reduce PITS by about 50 to 70%. You see a massive loss of the axons in this wing nerve. And that is quantified here, where you go from 260 to about 80 after 25 days. So there is a very severe defect here associated with the loss of PITS, the homologue of IRF2-BPL, when you do that in the wing nerves. Now, one of the interesting features was that we discovered that when you knock down in the neurons the protein using an RNAi, you see that there is an upregulation of wingness. This upregulation of wingness is about 2.5-fold. Interestingly, when Paul and Debdeep started studying drugs that are used in cancer to reduce wingless signaling, they noticed that there was a very nice suppression of the climbing defect that was observed in these flies. This clearly argued that indeed there is an upregulation of wingless signaling, and this upregulation of wingless signaling is really detrimental to the nervous system. Now, we know very little about wind signaling in the adult nervous system, but this clearly suggests that upregulation of wind signaling in the adult nervous system is toxic, and suppressing that is beneficial. So we then collaborated with Nancher Yeo and Matt Might at the University of Alabama, who made a zebrafish knockout for IF2-BPL and showed, I should say in zebrafish, there are 4 homologs. So in flies, there's 1, in human, there's 3, in zebrafish, there's 4 genes. And RF2-BPL is 1 of 4. And when they made a knockout, unlike in Drosophila, these fish are viable, but they're not. They have a diminished locomotor activity, and their startle response is affected. So normally they have a very strong startle response when you turn on, put them zebrafish in the dark, and these zebrafish do not have a normal startle response. Interestingly, these fish also show an upregulation of wingless signaling, as is quantified here. And again, when they use the same drugs that we had identified in the fruit fly, they show that there is a decrease with these drugs of LEF1 messenger RNA, which is obviously a key factor involved in the transcription of Wnt activation. So when they did this experiment and looked at the behavior, they noted that although the basal diminished locomotor activity when used these wingless inhibitors is not affected. The startle response was clearly improved when they used these drugs, suggesting again that Wnt is upregulated, and Wnt is having a negative effect on the function of some of the nervous system, and this can again be suppressed. So Wnt inhibition partially ameliorates the startle response in these fish. Finally, this raises a question, since we know relatively little about Wnt signaling in the adult nervous system, is it sufficient to overexpress the wingless protein in the fly, and does it cause an effect? And as is shown here, when we overexpress a LAC-Z and look at the nerve of the wing, we don't see any phenotype. But when we use wingless, and we need to do this for a protected period of time, after 5 days, there really is no phenotype, but you need to wait till day 15 to 20 before you start seeing a strong phenotype. And this is at day 25. And you again see that in this particular case, there is a very strong effect. Interestingly, an increase in Wnt protein was also observed in the patient cells. So we have patient cells, and in collaboration with Catherine Meyer at Nationwide Children, we've done experiments, and she has shown that indeed the Wnt protein is also upregulated in the cells that are derived from these patients. So in summary, then, for this portion of the story, PITS and RF2-BPL decrease leads to increase in wingless signaling, and the reverse, an increase in PITS and RF2-BPL, lead to a decrease in Wnt signaling. So drugs that decrease Wnt signaling suppress the neurobehavioral phenotype. Overexpression of wingless induces the same phenotype as the loss of PITS. And PITS and RF2-BPL physically and genetically interact. I didn't show you this data with CK1 alpha, which is a key player in the degradation of beta-catenin. And overexpression of CK1 alpha suppresses the phenotype induced by decrease of PITS in neurons. So clearly putting this genes in the wingless signaling pathway and playing a role somehow in the degradation of the beta-catenin, but also playing a role probably in some other aspects because the protein is in the nucleus, and this suggests that there is a nuclear component to the Wnt signaling, and in fact CK1 alpha is also in the nucleus, and so we're pursuing that. So I want to now turn to the last story, which is yippee-like 3 in zebrafish. This was a case that was presented by the Undiagnosed Disease Program, which is the program that was initiated at the NIH prior to the Undiagnosed Diseases Network. The Undiagnosed Diseases Program is an NIH program, and they identified a patient, a 12-year-old female that had symptoms at birth and was hospitalized with hypertrophic nerves, cerebral hypomyelination, so abnormal peripheral nerve conduction, hypotonia, and a series of other phenotypes that are shown here, including an abnormal eye and retina, abnormal cornea, and mild dystrophic features. This project was pursued by the MOSC, more specifically the zebrafish MOSC, by Monty Westerfield, John Postlethwaite, and their postdoc who did most of the work, Bernardo Blanco Sanchez. So this is a single individual, is this carrying a de novo mutation in this gene, and when they performed imaging, they noticed that this girl had massive hypertrophy of the peripheral nerves when compared to normal individuals. So here you have the sciatic nerve, and you have pretty condensed sciatic nerve, and here you have this vastly broadened sciatic nerve. This was observed not only in the sciatic nerve, but in other nerves as well. And so they found a mutation at the UDP, a de novo mutation in this yippee-like protein, which is very conserved. I think the conservation between zebrafish and human is 90%. And it was known to be a positive regulator of growth arrest, but it had not been really been shown to play any role in the nervous system or to play a role in development. And there's no known associated disease. They induced through CRISPR 2 mutations that are very similar to what is observed in the patient. So they induced through CRISPR 2 mutations, and both of these mutations would lead to an early truncation of the protein. And to our surprise, when they looked at the YIPI-like 3 knockouts in the zebrafish and stained for acetylated tupilin to label the axons, the peripheral nerves were hypertrophic in this mutant. And so this obviously provided a very good model already for what that indeed these mutations in YIPI-like 3 proteins are the cause of the disease. They then went on to show that indeed the gene is expressed at the right time, in the right place, and is expressed in the CNS, in the regions of the developing motor nerves, as well as the glial cells early on. And when they looked at the Schwann cells, they noticed again that they are hypertrophic, that clearly the neurons are defasciculated, and they're hypertrophic, and there is also a key problem associated with the maintenance of the Schwann cells. And so when they looked at myelin basic protein, there was, even though there is a hypertrophic nervous system, there was almost a complete loss of myelin basic protein in these mutants. So this then suggests that there is a key problem with the Schwann cells, and in fact also the oligodendrocytes in this mutant, and that there is a differentiation problem of these cells in these mutants. Interestingly, they went then on to do metabolomic studies on these mutants and found that in the PNS there was an upregulation of phosphatidylic acid, which is known to cause a problem with Schwann cell differentiation. So somehow this gene affects lipid metabolism during differentiation of the oligodendrocytes and Schwann cells and perineural cells. And they showed that the ceramides that are longer than C24, which are key lipids that are involved in myelin formation, are upregulated, and that is known to cause a decrease in oligodendrocyte differentiation. So in summary, then YIPI-like 3 is a determinant of glial cell development. I'm not sure the word determinant is the proper word, but it's a key player in the differentiation of the Schwann cells and the perineural cells and the oligodendrocytes. And it's required in the immature precursor population, and in the absence of YIPI-like 3, you have clearly a lack of differentiation of the glial cells, and that probably leads to a problem with the axons. They defasciculate, they swell, and they become larger. So in essence, the zebrafish, it shows you how nice the zebrafish model is here to make a direct connection with the phenotypes that are observed in the patient. There is a decreased maturation of oligodendrocytes. There is a defect in Schwann cell and perineural glial cells. The zebrafish model obviously validates that YIPI-like 3 is the cause of this human disease. And finally, it's interesting to note that in the discussion, the authors kind of discussed that YIPI-like 3 is a negative regulator of Wnt signaling, which is exactly what I was showing you for IRF2-BPL. And it wasn't shown that this is a major player in the differentiation of the perineural glia, Schwann cells, and oligodendrocytes, but obviously that's an interesting aspect to pursue. And I just want to give an end here with the credit for the zebrafish team that consists of Bernardo and some of the members of the Monty Westerfield and John Postlethwaite's lab at the University of Oregon, the Undiagnosed Disease Program at the NIH, and the Pacific Northwest National Lab, which was part of the model, which was part of the UDN, who did the metabolomic studies in zebrafish. And finally, I need to acknowledge a lot of people. There's no way I can acknowledge all the people. I already mentioned the key players for each of these projects. They are listed here. We participated in Marvel, and you'll hear another story about Marvel. The technology is a key player here, the CRIMIC and drop-in technology developed by MOSC and screened by Peitang, the IRF2BPL that I already mentioned, the MOSC for the undiagnosed diseases network. And I really want to acknowledge Shinya Yamamoto and Michael Wangler because they've played a really important role in the Drosophila MOSC here. And finally, you know, many of the clinicians, especially clinicians at Baylor, but obviously the clinicians throughout the UDN network. This is just to give you a sample as to how many people are involved at one site, in this case, Baylor. And finally, I want to acknowledge Jim Lipsky, who's as the head of the Center of Mendelian Genetics, together with Jen Posey here at Baylor, have really helped us in the discovery of many human diseases. So I'll end up here and be glad to take questions. I'm sorry I went over time, but for those of you who are interested, I'll be glad to take questions. All right. I only see posting from Jessica, so I assume that there is no questions here. Here's a question. Can modifiers for complex disease be done using model organisms? The answer is theoretically yes, although we have not tried that. I would say that the modifiers, obviously, the simplest way to do that is, for example, you have a de novo disease in a patient and you have one mutation, but you have another de novo locus. You can model those two in Drosophila and see if there is a synergism. And we've done that for some genes, and in some cases that's positive. And the answer is yes, you can therefore look at modifiers. But obviously it becomes quite tedious to do that. And certainly doing screens for modifiers would be very tedious. But if you have candidates from the human genome, then that becomes feasible. Regarding Nedimus, what about the role of LRP5? To be honest, I have no idea if LRP5 is involved. And so I don't know if it does play a role or not. As far as I know, we have not explored this, but I would have to refer to Paul and Deb Deep about this. So the next question is from Eric Lai. Hi, Hugo, you mentioned that there are 2 TNPO homologs, and does the other one rescue the fly mutant, or is it something about the levels that is an issue? We don't know. We have never tried to do another rescue. We try to rescue it with the human gene and we fail. We do rescue it with the fly gene, but we don't know why there is no rescue, and we didn't attempt to do other TNPO homologs and screen for those to see if they rescue. So the answer is we don't know. Then Matthew de Cruz asked another question. When considering introducing human genes into a model organism, if it chose to codon optimize to get a better translation of the protein, how do you think the scientific community would respond to the human sequences being altered from the original sequence? I don't think this is a problem at all. We've done humanization. We've done codon optimization. In general, codon optimization has not been an issue. I think it probably is an issue occasionally, but in general, codon optimization is not a problem, and there is no ethical concerns as far as I know to do codon optimization and put them into fruit flies. Next is Kingston Stephen Lee. Hi, Hugo. I'm wondering if you have an insight of using model organisms to screen drugs. Yes, as I mentioned and showed, we have identified drugs for ACOX-1, and We have identified drugs for IF2BPL, and we've identified drugs for other genes by doing screens in fruit flies. Essentially, you work out the molecular mechanism, and then you focus on trying to understand what's going wrong. Like in the case of IF2BPL, you screen and use drugs to suppress the phenotype. And in this particular case, the drugs worked in flies and zebrafish. So yes, we can do this. And in fact, for ACOX-1, we applied to the Food and Drug Administration based on the observation that N-acetylcysteine amide, which is a drug that's being used already in human, could be used for patients, and we're waiting for the outcome. We've done that before for other drugs like the cypramine in other disease, and that they were approved. So, you use FDA-approved drugs, that makes the threshold for going into the clinic very low. And if the FDA approves the use of your drug for compassionate use, you can integrate them into the clinic really fast. Paul Marcoglisi answers about LRP5 here. He was the one who did the work. As you can see, is not checked in screening. What we have found is a more direct action of IRFB on the Wnt ligand, and that's what we've pursued. Pael Ganguili, for instance, if a particular variant behaves as a loss of function in eyes and wild type like in the wing, how do you explain such tissue-specific differences between variants? Also, is one type of assay enough to capture the entire range of variant dysfunction? The first question can be answered. Obviously, in the case of transportin, you probably have 50 or 100 different cargoes or maybe more. These cargoes are different in different tissues. If you affect, let's say, with an amino acid, the cargo binding domain, you may have a different phenotype depending on where you and how you affect the cargo binding domain, because not all the portions of that domain interact with the same protein. So you're going to have some specificity in phenotype because you have so many proteins that rely on transportin to get into the nucleus. The second question is one type of assay enough to capture the entire range of variant dysfunction? And the answer is no. Ideally, you'd like to do different assays, and that's why we did overexpressions for the case of transportin in different tissues to see if we had consistent phenotypes. And in the case of transportin, the phenotypes were pretty consistent. We've done the same for other genes like GDF11. And in that case, we also saw somewhat consistent phenotypes in different tissues. So I think, it's fair to say that you can do many different assays, but in the case of transportin, when you have 15 patients and you clearly have some association with the proteins domains that is consistent, you have rather compelling evidence to move forward. All right, well, I see that Jessica sends another post to look at the other seminars that I advertised in the talk. And then John Abrams, can you elaborate on the deliberations that determine the choice of which model to use for functional studies, fish, worms, or flies? I think the answer to that is not really. I can, in the sense that what we do is we have the people that run the model organism screening centers look at these variants. They look at them in many different species. They look at the reagents that are available in all the different species. They look at, is the variant really a decent candidate for this disease? They have a meeting every month on Mondays. In fact, I missed the Monday meeting because I was preparing my seminar, but today there was a meeting where all the physicians that submit the variants together with the people that run the model organism screening centers at WashU and at Baylor and at the University of Oregon team up together and discuss the cases with the physicians and decide, obviously, if the gene is not conserved in worm and flies, it's automatically a fish gene. If the gene is conserved in worms and flies, in worms, typically they like to model genes that are conserved where the amino acids that are variant in the patient are conserved into C. elegans, and so mutate those into the C. elegans using CRISPR. All the other cases essentially go to the flies. If we can do it, and if, you know, we will create the CRISPR reagents typically, and then see if it's worth pursuing at a later stage. All right, it looks like that was all the questions that I saw. And if you have any questions or any concerns, you're more than welcome to fire up an email, send it to me, and we'll try to help you. I encourage you to participate in the other talks so you hear about all these databases and information that's available. There is a wealth of information to be mined, and typically as model organism researchers, we're not very well tuned to that kind of databases, and getting a tutorial about them might be helpful for many of you. Thank you.